Hey everybody, welcome to our first lecture on the urinary system. In this video and the next, we're going to be talking about the four organs in the urinary system, including the two kidneys, which are always making urine, the ureters, which are carrying urine down to the urinary bladder. The urinary bladder, of course, is storing the urine, and the urethra empties the bladder, carrying the urine out of the body. So those are the organs we're going to discuss. And as we talk about the kidneys, we're going to be aware of its major functions, which include filtering the plasma and removing waste from it, regulating blood volume and blood pressure, regulating the osmolarity or concentration of blood as well. And those are my big primary functions. My secondary functions include the production of renin. Renin raises blood pressure and is an important thing to help regulate blood pressure. The kidney also makes EPO, which we know stands for erythropoietin, which stimulates erythropoiesis or the production of red blood cells. Kidneys also regulate pH and form an important hormone, which is necessary for calcium homeostasis, called calcitriol. Okay. Before we get into the kidneys, I want to briefly take a look at their neighbors. So sitting on top of each kidney is an adrenal gland, also known as a suprarenal gland, making a bunch of hormones in both the cortex and the medulla. So the cortex makes hormones like aldosterone and cortisol. The medulla makes hormones like epinephrine and norepinephrine, and there are other hormones from this gland as well. All right, let's look at some kidney anatomy. This is a frontal section of the kidney, and we see on the very outside there is a fibrous capsule, which is connective tissue that is going to help support and protect the organ. Then we got two major regions. This outer region that my cursor is dancing around in is the renal cortex. And remember, that is where urine is going to be made. Then deeper to the cortex, we have the medulla. And the renal medulla is made up of all of these separate renal pyramids. Now notice in between the renal pyramids, we've got these areas that look like cortex, and they're basically cortical tissue. And these are called the renal columns. Those are the renal columns. A pyramid and its surrounding cortical tissue, including the columns, is called a lobe of the kidney. Now remember that word lobe. We're going to come back to that in a minute. So we had the cortex. That's where urine is always being made. Urine is actually flowing through ducts in the pyramids. And the tip of a pyramid is called a renal papilla. And urine is always dripping out of a renal papilla into this little tube here called a major calyx. Sorry, a minor calyx. My brain is 2 steps ahead of my mouth here. That's a minor calyx. And if you look over here, this is a minor calyx, and this is a minor calyx. And when minor calyces join together, we get a major calyx. And then when all the calyces have joined together, and we've got this wide funnel-shaped tube, that is called the renal pelvis. The renal pelvis is going to exit the kidney and become the ureter. And the indentation where the renal pelvis is exiting and also where blood vessels are coming in and out is called the renal hilum. And the space occupied by the calyces, the pelvis, and the blood vessels and the adipose tissue here is called the renal sinus. So we got a lot of good kidney anatomy right there. And in fact, we can start a basic pathway of urine flow right now. So urine is formed in the renal cortex, goes through the pyramids, drips out of the pyramids, drips out of the papillae into the minor calyces, into the major calyces, renal pelvis, ureter, urinary bladder, urethra, toilet. So that is a very basic pathway of urine flow. We'll expand on that later. Another thing that we got to do that's super important is renal blood flow. The renal blood flow is important because the kidneys are essential to blood homeostasis. The kidneys are getting about 25% of your cardiac output every minute. And that is a huge amount of blood for the size of these organs. So let's take a look at the gross vessels that we can see here. Starting with our renal artery, we know the renal artery is going to branch off the abdominal aorta. As the renal artery approaches the kidney, it is going to branch into some segmental arteries. Segmental arteries then branch to give us interlobar arteries. Notice our interlobar arteries are coming up between the pyramids. And remember, the pyramid and surrounding cortical tissue was called a lobe. So that's why these guys here are interlobar. The interlobars then branch to give me these arteries that curve over the base of the pyramids. And those arteries, because of their curviness, are called arcuate arteries. And then arcuate arteries give off multiple branches up into the cortex, which are called interlobular arteries. Now that's it for the gross blood vessels we have here. We'll look at the microscopic blood vessels in a second. So microscopic blood vessels are going to come off the interlobular arteries. And eventually those microscopic blood vessels, that microvasculature, will empty into interlobular veins. And we see these blue guys here are interlobular veins. Interlobular veins empty into arcuate veins. Arcuate veins become interlobar veins. And then this is kind of weird. As the interlobar veins join together, they keep their name. They're still called interlobar veins. So we actually don't name them segmental veins, although that would be perfectly logical. Just traditionally we don't do that. And then here we have all the interlobar veins eventually uniting to make the renal vein. So those are my gross blood vessels. If we look at the microscopic vessels, we can see the connection between the interlobular artery and interlobular vein. Now here's my arcuate artery right here. Coming up into the cortex off the arcuate artery is an interlobular artery. Notice that the interlobular artery is giving off this guy right here. This is called an afferent arteriole. It leads into a ball of capillaries called the glomerulus. That's where blood is going to get filtered, and the filtered blood enters this network of tannish tubes here. The blood that doesn't get filtered, which is still about 80% of the blood volume, ends up in the efferent arteriole, which is going to give rise to either some peritubular capillaries, which are these guys right here, or a group of vessels called the vasa recta. And then these capillaries end up emptying into an interlobular vein, which we see over here, then onward to an arcuate vein, then an interlobar vein, and a renal vein. So now we've got the basic flow of blood. And what we can now do is look at what's actually going on with the blood vessels and with the other tubes in the kidneys. All right, check out the afferent arteriole here. We know it's coming off the interlobular artery. It's coming into this ball of capillaries called the glomerulus. And it's in this ball of capillaries that we filter the blood. And we create a fluid called filtrate, which is basically water and small stuff. And that filtrate flows through these little tubes in the kidney. And as the filtrate flows, it gets modified. Now, of course, not all the blood, like not 100% of the blood ends up turning into filtrate. Only about 20% of the volume does. So the other 80% continues on into the efferent arteriole and ends up in the peritubular capillaries or in the vasa recta. And between these capillaries and the renal tubules, we're going to do exchange. If there's something in the filtrate that we want to keep in the body, we reabsorb it, meaning we move it from the fluid here back into the blood. If there's something that was was not filtered, but it's a waste that we want to get rid of, we secrete it, meaning we take it from the blood and add it to the filtrate in the renal tubule. And we do that in multiple locations. And all this reabsorption secretion turns the filtrate into urine. By the time we're done in these tubules here, and we end up in this collecting duct, we now finally have urine. So these are the 3 steps of urine formation. Now, one of the important things to realize is that by regulating the amount of water we reabsorb, we can regulate blood volume. So remember, in reabsorption, we're taking stuff from the filtrate, putting it back into the blood. And we can adjust how much water we reabsorb. If we reabsorb more water, we pee less. If we reabsorb less water, we pee more. And by adjusting our volume of urine output, we're thereby regulating blood volume and blood pressure. Similarly, we can regulate the chemicals we reabsorb, meaning we take them back into the blood, or the chemicals that we secrete, meaning we add them from the blood into the filtrate. So if we regulate what chemicals we reabsorb and secrete, and to what degree, we're going to regulate our blood chemistry, our blood electrolyte concentration, as well as our blood pH. So this is the main way in which the kidneys are going to affect homeostasis of the blood and thereby homeostasis of the body. All right, those processes, those 3 processes of urine formation occur in the structure called the nephron. There are about 1 million nephrons per kidney. Each nephron has 5 parts. It has a glomerulus, which is the ball of capillaries we see here. Note it is surrounded by a glomerular capsule, which then leads into a next door twisty tube called the proximal convoluted tubule. Then we get a down and up tube called the loop of Henle. Then we get a farther away twisty tube called the distal convoluted tubule. And that empties into a collecting duct. So the collecting duct is not technically part of the nephron. Now, this is where everything is happening. This is in the nephron. It's where I do the 3 steps of urine formation. I'm going to do filtration, reabsorption, and secretion. Now, if we look at a nephron again, we can notice the vasculature associated with it because, of course, reabsorption and secretion require exchange between the filtrate in the renal tubules and the blood in neighboring blood vessels. All right, let's talk about that glomerular capsule for a second. So the glomerular capsule encloses the glomerulus. The glomerulus is the ball of capillaries here where filtration happens. The glomerular capsule is going to help the filtration occur, and it's going to receive the filtered blood, and that filtered blood is known as filtrate. And notice how we have an inner and an outer layer of the glomerular capsule, which is still sometimes called the Bowman's capsule, which is its old-fashioned name. Notice also how the glomerular capsule empties into the proximal convoluted tubule. Now, like I said, the glomerular capsule has 2 layers, an inner and an outer layer. The inner layer is called the visceral layer, and it is made out of these light purple-looking cells here called podocytes. So this is one podocyte right here. There's his nucleus, his cytoplasm, I see his membrane, and his many extensions. The extensions of the podocytes are what gives these cells their name. Podo means foot or feet, so these cells have all these little feet coming off them. And notice how the feet are kind of interlocking or interdigitating, leaving a little space between, and that is going to contribute to the filtration. So this visceral layer helps filter the blood. Now, the outer layer is the parietal layer. It is made of simple squamous epithelium. It is going to help create the lumen for that glomerular capsule, and the filtrate is going to be contained by this parietal glomerular capsule, this simple squamous epithelium right here. Okay, here we see the real thing. This is a glomerulus plus podocytes. This is the lumen of the glomerular capsule, and this pinkish line right here is the parietal glomerular capsule, which of course is made out of simple squamous epithelium. Here we see another one. Make sure you can identify the glomerulus, the visceral glomerular capsule, as well as the parietal glomerular capsule. Now, coming out of the glomerular capsule, we got the proximal convoluted tubule. Proximal would be proximal because it's close to the glomerular capsule. Convoluted because it's very twisty. This is the major site of reabsorption secretion. So this guy would be covered by peritubular capillaries. We're reabsorbing stuff from the filtrate in here into those capillaries and then secreting stuff from the capillaries into the filtrate. Now, the twistiness gives us more time and more surface area for reabsorption secretion. The epithelium of the PCT here is simple cuboidal. We see some of the cells right here. Notice that they have microvilli, which further increase the surface area. These cells of the PCT are also rich in mitochondria because reabsorption and secretion are energy-dependent processes. So we need a bunch of ATP, so we need a lot of mitochondria. The proximal convoluted tubule empties into the loop of Henle, also called the Nephron loop. It is going to be a little complicated in its physiology, but overall, it's going to help the collecting duct reabsorb water. And we'll talk about how that happens a little bit later. Now, the distal convoluted tubule is distal because it's farther away. It's convoluted because it's twisty. It is where I am going to further do reabsorption secretion, primarily like on a need-based manner. So like if I'm low in sodium, I'll reabsorb sodium. If I'm high in potassium, I'll secrete potassium. And hormones usually tell the DCT cells, like how much reabsorption and secretion to do. It is a simple cuboidal structure, but there are fewer microvilli and fewer mitochondria because of the fact that we're doing less reabsorption and less secretion. Now, the collecting duct is going to get urine from multiple nephrons. Collecting ducts run down through the renal pyramids, in other words, through the renal medulla. They're actually going to combine to make bigger and bigger ducts, eventually emptying into a minor calyx. The collecting duct is mostly simple cuboidal as we're up towards in and towards the cortex, and it becomes simple columnar the farther into the medulla we go. And the collecting duct doesn't do any secretion, but it does do some water reabsorption if we're dehydrated. And there are hormones that'll tell it to do that. So it'll reabsorb water if we're dehydrated. All right, let's talk about the blood vessels associated with the nephron, starting with the glomerulus. It is this ball of capillaries here. We see it covered by podocytes. They're fenestrated capillaries, which is going to make for good filtration. The glomerulus is weird in that it has a really high blood pressure for a capillary, and the high blood pressure is created because of the size difference between the afferent, this is the afferent arteriole, and this is the efferent arteriole. The afferent is much bigger, so it's easy for blood to get into these capillaries and harder for blood to get out. So that raises the pressure in here, and that high pressure drives filtration. If we look at our electron micrograph over here, so this is the real glomerulus here. Note the width of the afferent arteriole compared to the width of the efferent arteriole. All right, the peritubular capillaries, like their name suggests, peritubular, around the tubules. They surround the renal tubules, basically the proximal and distal convoluted tubules. And these capillaries are getting blood from the efferent arteriole, and we see them emptying into the interlobular vein. Now, peritubular capillaries, because they're around the convoluted tubules, they are, of course, involved in reabsorption and secretion. In fact, they've got a low blood pressure and a high osmotic pressure, and that really helps them reabsorb water. Remember, osmotic pressure will draw water towards it. So a high osmotic pressure helps draw water into these capillaries. The vasa recta are these capillaries enveloping the loop of Henle, and the vasa recta is going to kind of work with the loop of Henle to help the collecting duct reabsorb water from the urine if we need to. And notice that the vasa recta here is getting blood from an efferent arteriole and then emptying into an interlobular vein. All right, so we know we got our 3 steps of urine formation: filtration, reabsorption, and secretion. And I want to talk about how those 3 steps occur. So let's start with talking about filtration. And we know filtration happens in the glomerulus. And we know the glomerulus is a ball of capillaries. The type of capillaries is fenestrated, and we can see the little pores, the little fenestrations. And little holes are what makes a good filter. A good filter is going to have small holes, and fenestrated capillaries definitely fit that bill. Now, this is a podocyte sitting on top of the capillary, and you can see its extensions, its feet, which then branch and have further extensions, and we can see how the extensions interlock or interdigitate, leaving little tiny spaces called filtration slits in between them. And so this also helps in the filtration process. And the podocytes plus the fenestrated capillary make up my filtration membrane. And the general idea is water and small stuff can get pushed through the fenestrations and through the filtration slits and end up in the glomerular capsule as part of filtrate. Notice how formed elements, white blood cells, red blood cells, and platelets are all too big. Large proteins are too big as well. All right, so that is the filtration membrane. Another thing we got to consider is the filtration. There are actually 3 pressures impacting filtration. There is the hydrostatic pressure, or the blood pressure of the glomerular capillaries, which is about 60 millimeters of mercury. That's high for a capillary because remember, afferent being big, efferent being small, creates high pressure in here. Now, this outward pressure is slightly offset by the hydrostatic pressure of the filtrate, trying to push its way back in. That's about 18, as well as the osmotic pressure of the blood. Remember, the blood has its own osmotic pressure due to albumin, and that's going to try and pull water back in. So notice we got 60 millimeters of mercury of pressure going out, 32 plus 18 going back in. 32 plus 18 is 50, 60 minus 50 is 10. So we get a net pressure of 10 millimeters of mercury pushing water and small solutes out. And this is actually a really high net filtration pressure, much higher than what we find in regular run-of-the-mill capillaries in, other organs, skin, muscle, liver, etc. And this high filtration pressure results in the production of a lot of filtrate. Now, of course, you might be wondering, how much filtrate? Well, we make about 125 milliliters per minute. And that is called the glomerular filtration rate. And it is very dependent on that glomerular blood pressure. And I really want my glomerular filtration rate to be very constant around this number, because if I make filtrate too quickly, the filtrate moves too fast and I don't have time for reabsorption. And if I make filtrate too slowly, the filtrate flows too slowly and I start doing too much reabsorption, including reabsorbing bad stuff. So I want it to be just right. And one of the main ways I am going to maintain my glomerular filtration rate is something called the myogenic mechanism. Myogenic means created by muscle, And it's due to the smooth muscle in the afferent arteriole. If systemic blood pressure drops, so like body-wide blood pressure drops, I don't want blood pressure in my glomerular capillaries to drop. So what's going to happen in response to this drop in systemic blood pressure, my afferent arteriole is going to dilate. That lets more blood in the glomerulus, which is going to then maintain the pressure there. And if I maintain the blood pressure here, I also maintain the glomerular filtration rate. Similarly, if blood pressure in the whole body rises, I don't want it to rise in my glomerulus. To prevent it from rising in my glomerulus, what I'm going to do is I'm going to constrict my afferent arteriole. So if systemic blood pressure rises, the afferent arteriole constricts, less blood gets in the glomerulus, and we maintain the pressure there, thereby maintaining the glomerular filtration rate. Okay, another thing is called the tubuloglomerular mechanism, and let's run through a sequence for that. If glomerular filtration rate increases, the filtrate starts to flow quickly through the nephron. There is a part of the nephron in the distal convoluted tubule called the macula densa, and if filtrate is moving too quickly, the macula densa is going to sense that and release a local signal that's going to constrict the afferent arteriole, which means less blood is going to get in the glomerulus, which is going to bring glomerular blood pressure back down, which is going to bring GFR back down. So this is called the tubuloglomerular mechanism, and it's also important. So this mechanism and the myogenic mechanism, they're always working to maintain glomerular blood pressure, which then maintains glomerular filtration rate, which is crucial for homeostasis. Okay, another thing we got to discuss is what the sympathetic nervous system does to glomerular filtration rate. Remember, sympathetic nervous system is your fight or flight system. And if we get an increase in this sympathetic activity, we're going to have a bunch of norepinephrine and epinephrine released in the body. And that's going to get the afferent arteriole to constrict. When the afferent arteriole constricts, less blood enters the glomerulus. When less blood enters the glomerulus, glomerular blood pressure and thus glomerular filtration rate fall. So if we're making less filtrate, we're going to end up making less urine. And by making less urine, we're keeping blood volume up. And by keeping blood volume up, we're also keeping blood pressure up, which is crucial to maintain blood pressure and blood volume during a fight or flight event, because we need to have the blood pressure high to send blood to our working muscles, to our lungs, to help us deal with that threat. All right, let's call it quits on urinary system video number 1 here, because we're about a half hour in. We'll pick up next time with the renin-angiotensin system, and we'll finish up the kidneys. Then we'll talk about the ureters, the urinary bladder, and the urethra. We'll talk about peeing, and then we'll be totally done. So I will see you next time for urinary system part 2. Bye-bye.